So you can see the topic, today's topic, apoptosis, its description, the mechanism of apoptosis and its significances. It is in your competencies under the domain of general physiology. So I will restrict my discussion only on this. So let us start with apoptosis. Apo means fall off. Apoptosis means fall off. Means death. And this death is not an ordinary death. It is a programmed cell death. Decided. There may be some death of cell because of injury. So this is not a programmed cell death. This type of death is called necrosis. Some bacteria has invaded into my tissue and through inflammation it destroyed some tissues. This is not a programmed cell death. This is necrosis. So necrosis, every cell has to die. Every cell has to die. Either because of necrosis or because of programmed cell death. So we will be discussing today this programmed cell death. Is it normal dying of cells by apoptosis? Is it going to help us? Is it necessary for our survival, or it will lead to our death? So we'll find the need of apoptosis, for the organism, or the importance of apoptosis in our body. Apoptosis, cell death occurs by two means. One is by, as I said, one is by necrosis, another by apoptosis. Apoptosis results from two stimuli. One is internal stimuli. Within the stimuli comes from within the cell. Another stimuli comes from outside the cell. So their stimuli are of two types, internal and external. Whereas the cell death, it is caused by exposure, mechanical damage, exposure to toxic substances, lack of oxygen, extreme temperature and so on. Okay? So see the differences between necrosis and apoptosis. What is the differences? It is always pathological and it may be physiological as well as pathological. It affects adjacent cells, but our apoptosis will not disturb the neighboring cells. Only this cell will die. Others surrounding them will remain intact. Cell size here is increased, but here cell size is gradually, the cells will form bubble-like structures. This is called bubbling, we'll see. Through bubbling, there will be necrosis of, there will be degradation of the nucleus, degradation of mitochondria, degradation of all internal organelles, breakdown of membrane. So as a result, we will get apoptic vesicles, and these apoptic vesicles will be engulfed by phagocytes, macrophages, and they will be out of my body. So here The necrosis is passive. Here it is the active process. Lot of enzymatic reactions will be happening. And causes inflammatory reactions? No inflammatory reactions. So these are the differences between apoptosis and necrosis. Bivalve's equation or MCQ or short question. See, this is the normal cells, and under necrosis the cell size is increasing. Thereby blasts are formed like pseudopodia, and it invites inflammatory reactions. So these are the lymphocytes. They are the lymphocytes. So they attack these cells, and as a result the cells is degraded, lost. This is necrosis. But here you see what is the process? Here cell size is decreasing, and there is formation of bubbling, and ultimately this will be fragmented like this, apoptic bodies, and these apoptic bodies will be engulfed by macrophages, and they will engulf it by the process of phagocytosis, and all the ingredients, complexes like protein, carbohydrate, fat, these are there. So everything will be digested by the lysozymes present within the phagocyte or macrophage. So they will digest them into units. See, if there is any carbohydrate type of mucopolysaccharides, say, so this will be converted into glucose and it will be eliminated. Eliminated into that can be reutilized, recycled. Proteins will be hydrolyzed into amino acids within the by the lysozyme. These amino acids will be released into the cytoplasm, they will be utilized further. So in this way, apoptosis is done. So as I said, it means falling off. Apoptosis, so this is the definition, and apoptosis is a normal phenomena. It's not an abnormal phenomena. It is a normal phenomena, and it is essential for normal development of the body. I will explain how it is essential. Apoptosis is a pathway of cell death which is induced by a tightly regulated suicidal program. Means this is genetically controlled. But that one is not genetically controlled. It is genetically controlled. Some genetic material will be produced, and that will cause either that will be produced by the external stimuli or by the internal stimuli. Then apoptosis will be leading. And apoptosis removes see the significance, one of the significance. See, what is this? It removes unwanted cells. It removes unwanted cells. It removes potentially harmful cells that may, like cancer cells, virally infected cells, cancer cells, degenerated cells. We have learned degeneration and regeneration of neurons. Didn't you learn in class 12? Degeneration, regeneration of nerves, nasal granules, nerves. What happens when an axon is cut in the somatic end or in the peripheral end? So a lot of reaction takes place. It is when a neuron is damaged during development, during embryonic stage. You see, when a baby is born, it's unable to move, unable to hold something. Why? Its central nervous system are not developed. They are developing. Some neurons already developed, they are dying, they will be dying, and new neurons will be formed. And while forming, and it will form, these new neurons will form synapses. Synaptic control, then only control curve will come. When an infant undergoes maturity, That means in the brain there is making lot of connections, neural connections we are making. Purpose to control an activity in a regulated way. So during this process of development, lot of neurons dies, new one is formed, and particularly this is essential during the formation of synaptic connections. Understood? So in that way also it is very much important. Self-reactive cells, sometimes we produce some cells through our immune process, and that cells destroy our own body cells, thinking that these cells are immune body, as it happens in lupus nephritis, lupus erythematosus. So there are several diseases like this. We call it autoimmune disease. Okay. So there are a lot of importance I will come into. So apoptosis removes unwanted cell potential. This is already, and this occurs. This cell death, which is apoptosis, occurs without causing stress or damage to the neighboring cells. I already told. This will not damage the neighboring cells. It's a physiological condition. Apoptosis is a physiological condition. Daily, lot of cells are dying, and lot of cells are producing. And to keep the balance, we are keeping balance. And the babies, their rate of destruction, rate of death of cells is less than the rate of production. That's why they are growing. And we are maintaining. I am not maintaining, I am reducing gradually. You are maintaining. After attaining certain age, you will be also losing. There will be more death than more production. So under normal condition, about 10,000 cells are formed daily, whereas same amount is dying. So it is maintaining homeostasis. So it is maintaining the balance. As a result, I am maintaining my body weight. You are maintaining your body weight. So this is important. So you see, that's why we call it a physiological process. There are some physiological processes I will come into. Now go through it. Try to remember all those points. Why these are important. For exam purpose also. Importance of, in your book, some points you may get, may not. So better you try to capture this. These are the importance. The purpose of apoptosis, first point is already discussed. Second point, it plays a vital role in cellular homeostasis. Oh, that one also I told. It is useful for removal of cell that is damaged beyond repair or affected by a virus. It's happening every day. You don't know your superior layer, external layer of epidermis is gradually going off, and some new are, but you are not noticing it. It happens there. In our intestine, intestinal mucosa, where there is papilla, the tip of the papilla, there are some endothelials, there are some secretory cells, mucous cells. They become, at the tip, some of the cells become isolated from the neighboring cells. And what is the use of their staying there? So apoptosis starts to destroy it, and in that place new cell is generated. While you were children, you had a thymus gland. Now you don't have, or it is atrophied. So how it became like this? How it is degenerated? By the process of apoptosis. The girls, after adolescence, there is a reproductive cycle, menstruation. In every cycle, there is shedding of endometrium. There is shedding of endometrium, and this is initiated by the newly formed endometrium again undergoes necrosis, and it is not necrosis, apoptosis, and this is because of breakdown of the small blood vessels, initially by the blood vessels, by apoptosis. And when the blood supply to the endometrial cell will be diminished, all the cells will be shedded off gradually. So it is also, we can see apoptosis there also. This point, it is responsible for removal of inappropriate clone of immune cells. Some clone cells are produced, but they are not effective. They are not proper. They are improper. They are going to damage. So, our internal or external environment will identify this. So, immediately this will by the process of apoptosis, this will be dying. As I said, large number of neurons during developmental process are dying, and for the developmental cell, new neurons will be formed, and they will, while it is happening, it will happen during formation of synapses. Some during our embryonic stage, when we develop our finger digits, fingers, they are interconnected by web-like structures. When a fetus grows, within the mother womb, these structures are removed by apoptosis. Unless these are removed or there is some difficulty, then you will find some fingers are, in some people fingers are joined, and some is short, some is big, some digits are big. So these are abnormality in apoptosis, in development. And during your development, in the embryonic stage, at one stage there is no sex difference in the fetus, and ultimately sex difference developed. So this is the example. Due to lack of apoptosis, it happens during embryonic life. These are the waves. These are the waves, red colored, joining the finger digits. And see, there are both the ducts. Okay, now they will be separated. When these ducts, red and blue colored ducts, are separated, this will form female sex, and the blue one will form male reproductive system. So this is done by, so here what is happening? Actually the red color tubes are undergoing death by apoptosis. Here blue color is going out by the process of apoptosis. As a result, and if just think, if both the tubes remain, what will happen? This is happening during menstrual endometrium. These are the endometrial tissue, and it is okay, normal. But at the time of before bleeding starts, so there is breakdown of here. Some blood vessels are there; they will be ruptured. So this region will not get blood supply. So they will gradually break down and fall out. This is actually happens during menstrual bleeding. And you see the thymus gland at young age in a children, but in adult, see the size of the thymus. Regression of thymus gland. It is done by, and if this gland, thymus gland, remains active, it will also create some problem which is unwanted at an elder age, at an adult age. And now we have seen the mechanism. Did you Take the, noted the importance, all the importance you should know. And now the mechanism of apoptosis. The mechanism of apoptosis has, as I said, there are two pathways. One is intrinsic, another is extrinsic. So there are two pathways, intrinsic and extrinsic pathway. What are the reasons of apoptosis? Why should a cell go for apoptosis? There are two reasons. Withdrawal of positive signals. Positive signals will allow the cell to survive. What are these positive signals? Lack of growth factors. So this lack of growth factors for neuron. So if it is there, there will be degenerative processes in the nervous system. It happens in diabetic patients. That is, one condition is called diabetic neurosis. So there is what? After suffering long period of diabetes, they suffer tingling sensation in the foot, in the lower extremity, in the extremities. And it is due to demyelination of the nerve fibers. And it happens, myelin sheet is also made by Schwann cells. So there will be damage of these Schwann cells, so there will be axons will be exposed, and this will invite certain sensations. Then I will feel there's something tingling, uneasy feeling. So this sort of thing, pain. So this sort of thing may appear. Interleukin 2. This is also very much essential. Without this, we will discuss in detail during our immunity class. So interleukin 2 is also very important, and it is also a growth promoting or a positive signal. And the receipt of negative signals. So suddenly I got some negative signals. And this negative signal or absence of positive signal will lead to apoptosis. What are these signals? These are I already told in our previous classes, mentioned several times. We are producing a number of oxidants which are destroying our cells. That's why we take antioxidants. So when these, like hydrogen peroxide, O2 negative, superoxide anions, so they are very harmful for the cell. They can destroy the cell. When there is increase in such oxidants within the cell, this is the stimuli, internal stimuli. So this will lead to apoptosis. Okay. Then there are some death activators who will be producing the death activators. When these are activated, the cell will be died by the process of apoptosis. So these are the reasons. Because of these factors, negative factors and positive factors, they can lead to apoptosis. As I said, there are two mechanisms, intrinsic pathway and extrinsic pathway. Intrinsic pathway, the stimuli will come from outside, and intrinsic pathway, the stimuli will come from within the cell. Okay. And in extrinsic pathway, the stimulus is received from outside, as I said, and both the pathways, in intrinsic pathway, stimulus arises from within the cell. Both the pathway lead to activation of an enzyme. This is the main person who will initiate apoptosis. What is this? Caspases. These are group of proteins, caspases, particularly caspases 8 and caspases 9 are very important. Whenever this will be activated, the cell will undergo apoptosis. And activation of this caspase, it may come from intrinsic path, it may come from extrinsic path. So ultimately, to initiate apoptosis, the presence of activation of this protein is very much essential. And these caspases are who are the source? The cell which is undergoing, who will be undergoing apoptosis. So within the cell these are present. Only it needs, when it will be activated, the cell will undergo apoptosis. When not, okay, it will survive. So normal cell, shrinking, membrane is blebbing, so bubble-like structures are formed, and there is, see, what is happening to the nucleus? It is, there is nucleus, within that nucleus there are chromatin. So this chromatin will be deranged and they will be condensed. And after that, nuclear membrane will be ruptured. As a result, what will happen? The DNA will be exposed. Within the nucleus will be exposed. Now the DNA will be fragmented. Then no chance of growing. No chance of, once you damage the nuclei, no chance of dying. You damage your brain. Nucleus is the brain of the cell. So it is damaged by this, by defragmenting the DNA. And after that, since nuclear membrane is also ruptured, so the contents of the nucleus will come out, will enter into the cytoplasm. And by this time, what is happening? Here, every will be separated, and this fragment will be produced. These are called apoptic body, and this apoptic body will be lysed by who will lyse it? Phagocytes. They will engulf and phagocytize. Who can induce? Who are the inducers? These are the factors that can induce apoptosis. DNA family factors, growth factor withdrawal, calcium, excess oncogenes. Oncogenes are cancer producing agents. Oncogenes, nutrient deprivation, toxins, UV radiation, gamma radiation. So these may induce apoptosis. We already discussed. So there are two pathways, as I said earlier, intrinsic and extrinsic pathway. Intrinsic pathway also called mitochondrial pathway. Intrinsic pathway also called, and extrinsic pathway, death receptor pathway. Did you ever heard the name ligand? Ligand, L-I-G-A-N-D. What is this ligands? You think of a hormone. One organ, one tissue, thyroid gland, pituitary gland, pancreas, testes, ovary, pituitary, or can I name hypothalamus? Can I name hypothalamus as an endocrine organ? Why? Pituitary gland, it produces, how many parts are there in pituitary? Anterior pituitary, posterior pituitary, and there is intermediate lobe, no? All are secreting some hormones. Anterior pituitary is secreting, can you name one? Then one anterior pituitary hormone. Right, growth hormone. This is formed within the anterior pituitary. But this hormone cannot be synthesized by your own. Pituitary forms this hormone under the command of another brain, another area of the brain, and that is hypothalamus. So hypothalamus will secrete some releasing factor, and this releasing factor, when it will come to the cells of anterior pituitary, will stimulate those cells and it will produce, then only it will produce growth hormone, otherwise not. And this, how it is coming? How this growth stimulating, this growth factor is coming, or somatotropic growth from somatotropic releasing factor. So this will be coming through hypothalamico-hypophyseal portal system. This is a vascular system having capillaries in both ends. They are called such capillaries. Such blood vessels are called portal valves. I will ask one day while I will be starting serious cardiovascular system. So I will ask what are portal system. So portal system, the vascular system, usually normally what happens? Then artery, it is producing branches, and ultimately will form capillary, and it is the arterial end of the capillary. And from this arterial end, there will be venous capillary, and this will join and ultimately form veins. So there is formation of capillary once. But in case of portal system, there will be formation of capillary twice. One capillary is originating in the hypothalamus. From hypothalamus, this venous blood system, when it is taking venous blood, carrying those releasing factors, and again it has to send these factors to the anterior pituitary. So there, the blood vessels will be ending through the formation of capillaries. So this blood vessel, hypothalamico-hypophyseal portal system, are having capillaries at both ends. One at hypothalamus, one at the other end at anterior pituitary. And because of this system, the anterior pituitary gets necessary releasing factors from the hypothalamus. And what about the posterior pituitary? ADAs. Are they produced in the posterior pituitary? Are they produced in the posterior pituitary? No. They are not synthesized there. They are released from there only. But they are produced in the hypothalamus. How they are coming there? Not by portal system. There is so there, we know nerve endings in synapses or neuromuscular junction, they secrete something. We call it neurotransmitters. We call it neurotransmitters. There is here also it is like neurotransmitters. By a neuron, these are synthesized in supraoptic and paraventricular nucleus of hypothalamus. How they will be coming? Not through the portal system, but by the axon. Through this axon, they will travel, and the axon terminal is in posterior pituitary. And from there it will be released. And it will act. It will be released and will pass through the blood to the target organs. And since this neurosecretion is not acting on muscle or another neuron, we don't call it neurotransmitter. Rather, these products secreted from these nerve endings at posterior pituitary, they are, after release, they are carried by the blood. That's why we call it neurohormone, not neurosecretion. So ADAs, remember this point, why we don't call it neurosecretion, we call it neurohormones. Understood? Anyway, so now my point is, this is one endocrine gland. It has produced one hormone. Now, this hormone will find a target cell. Who will be having, on which cell it will be acting? The cell must possess a receptor for this hormone, otherwise not. My cells here, I will not respond to ACTase because these cells are not having receptor for ACTase. Okay, so if they find a receptor, then they will couple with a receptor, with its specific receptors. That's why every hormone has a specific receptor. Where it will find the receptor, there it will show its action. Otherwise not. All the cells are not having such receptors. So there its effect will be absent. This binding of hormone with the receptors. Here, in our case, we will call this ligand. Ligands are the substances that act on cells through binding with their receptors, like hormone. So we can tell these substances, compounds as hormone. So actually all the hormones are ligands. So when a ligand, it may be other than hormone. So when a ligand binds with this, it will create some changes within the cell response. In this case, we will call such ligands that leads to apoptosis, such ligands we call death ligands. We will provide some examples. Death ligands. And these receptors, death receptors. When they will bind with this, the changes mean there will be changes in mitochondria, changes in nucleus. So, as a result, the cell will die. Will it this kind of apoptosis, is it internal or external, intrinsic or extrinsic? Obviously extrinsic. This is extrinsic. This is not intrinsic. So extrinsic pathway will utilize this. But intrinsic pathway will not utilize this. We will see how the intrinsic pathway works. Intrinsic pathway, as I said, it is also called mitochondrial pathway. That means it will start from mitochondria. It will start from mitochondria. What are the stimuli? Cellular stress, viral infection, increased concentration of oxidants, heat, radiation, nutrient deprivation, hypoxia within the cell, and increased intercellular calcium concentration, damage to DNA. These are the cause by which mitochondria will be affected. See how it is done? See, internal stimuli leads to damage to mitochondrial membrane, increasing its permeability. Note it. This is the pathway for intrinsic. So what happens? Internal stimuli damages the mitochondrial membrane, increasing its permeability. So a lot of things will enter. Permeability has increased, and this will lead to entry of within the mitochondria. There are various cytochrome enzymes. Within mitochondria, these cytochrome A, cytochrome B, cytochrome C, these are all oxidative enzymes. They are the enzymes of electron transport chain. Okay, since permeability changes, so some cytochrome C will be coming out. Some enzymes will be coming out from the mitochondria, will enter into the cell, cytoplasm. So, entry of cytochrome C into the cytoplasm. Now the cytochrome binds to APAF, another factor, and forming an apoptosome, and this will cause activation of caspase. As I said earlier, caspase. This will initiate apoptosis. This is essential. Ultimately, you have to produce this. Here it is caspase 9, but in that pathway we will get caspase 8. Here caspase 9 is formed, and then we will see what happens when this is formed. So you just watch what happens after this is formed. When it is activated, what happens? So increased mitochondrial activity causes release of protein cytochrome C and SMACs. SMACs means second mitochondria derived activator of caspases into the cell cytosol. So now the SMAC binds to AAP. AAP, it is always inhibits apoptosis. So it will bind with and inactivate it. So its presence will not allow apoptosis. So first it has to remove this. It has to deactivate it. So this cytochrome C, then cytochrome C combines with AP, then forms apoptosome. And this apoptosome will activate caspases. Then process of apoptosis begins. So intrinsic pathway, in short, this is the binding side, and this is released, apoptosome is released, cytochrome C, and apoptosis started. Extrinsic pathway. So could you understand this intrinsic path? So now, so your books, when we will go to your books, again, you will be having some problem in actually understanding this. Now you will be able to follow. If you go through your book today itself, then you will learn. Otherwise, again, same problem will be getting. You also study. Those who are having Shambulingam, books of Shambulingam, if you possible, it is nicely written there. So, better I will suggest you go to Shambulingam. Even the significance is well written there. But in Akajan, it is very brief, not properly explained. But in other book, Gupta is also good. Also good. But it is for easy understanding, Shambulingam is good. See, so this is external pathway. So death ligands you have seen. Death ligands like hormones. There are specific hormones that they will cause the death. So they will bind with the death receptors. Then it binding of this with this will cause activation of caspases 8. As a result, what will happen? There will be active formation of effective or active caspase 8, and this will cause PCD means this is other pathway, mitochondrial pathway. There are two ways by which 8 and 9 both can be formed. One is this path, another is path. DNA damage. This will generate damage in the nucleus. DNA will be damaged, no, and this will cause release of mitochondrial cytochrome C, and then it will activate caspases 9. And this 9 here will get 8, both together will call apoptosis. Program PCD means programmed cell death, apoptosis. No need. There is a short animation. You see, the ligand is binding. This is the ligand, and this is the death ligand. That is the death receptors. Now they bound together. Okay. Now internal within the cell changes is coming within the cell. So here is going to formation of what is this substance that initiates apoptosis? What is the main thing? Caspase. So there will be a lot of caspases are produced. And this will lead to the changes. Now let us see what are the morphological changes that is taking place within the cell. First, try to remember these steps. These are the steps actually. This has been activated, caspases 8 and 9. They digest the proteins of the cytoskeleton. Every cell is having cytoskeleton for maintaining its shape. So immediately it will break the cytoskeleton first. Kumur bhenge dilo. So as a result, the cell will be deformed. It will be shrinked. It will be shrinked. Then the loses the contact. When it is shrinked, it will lose its contact with the neighboring cells. So activation, damage of the cytoskeleton, shrinking of the cell, separating of the cell from the neighboring cells, this will be isolated now. Then what will happen? The chromatin reticulum that is present within the nucleus, this will be deranged or degraded and from a compact condensation. There will be condensation. And the nuclear membrane will be fragmented. As a result, what will happen? The mitochondria will come up from DNA. DNA will be splitted. The DNA will be coming out, and the DNA inside nucleus is cleaved into fragments. Then what happens? Nucleus breaks into chromatin bodies. Okay? The cell membrane breaks. Now the cell membrane breaks into pieces. Now finally, what is happening? Some contents of the cell and some part of the membrane will come together and form a vesicle. These are apoptic vesicles, and this will be this will undergo phagocytosis, and there will be ending of life of the cell. Is that clear? So you take up this photograph of this, first this. So these are the gradual stages.